The structural importance of plasma membranes in and around cells. All cells are surrounded by a plasma membrane. Eukaryotic cells also contain membrane-bound organelles, while prokaryotes do not. This essay will describe the structure of these membranes and detail how they are adapted to their function. The cell surface membrane is a plasma membrane composed of a phospholipid bilayer. The phospholipid molecules are very similar to triglyceride molecules, in which one of the three fatty acid molecules has been replaced by a phosphate group. The bilayer acts as a hydrophobic barrier that prevents the passive diffusion of hydrophilic species, such as glucose and amino acids, into the cell. Hydrophilic channel proteins are embedded in the bilayer, which provides a route by which polar substances can enter, either down a concentration gradient by facilitated diffusion, or against a concentration gradient by active transport. The relative movement of the lipid molecules together with the arrangement of proteins gives rise to the term the fluid mosaic model. The bilayer makes the structure partially permeable so that nonpolar and lipid-soluble molecules that are small enough are able to pass through the membrane by diffusion, but water molecules cannot unless they are very small. Some of the intrinsic proteins have gates on the top, which must be opened by the proteins changing shape. This is sometimes achieved by a molecule binding to it. For example, acetylcholine molecules fuse with the receptor sites on sodium ion channels in the membrane of a postsynaptic neuron, causing the channels to open, allowing sodium ions to diffuse in. Some protein molecules in the membrane act as carrier proteins, but only when a molecule of ATP binds to them and is dephosphorylated. This is the mechanism behind active transport and occurs to move molecules or ions from an area of low concentration to an area of high concentration. This involves the expenditure of energy released during respiration. It occurs during selective reabsorption in the cell linings and the kidneys. Active transport pumps glucose and amino acids from the epithelial cells, steepening the concentration gradient between the cytoplasm and the lumen, allowing more glucose and amino acids to diffuse into the cell. It also occurs in plant root cells, as ions are actively transported in, lowering the water potential, allowing water to enter the root hair cells by osmosis. The membrane can also control what takes place in a cell. For example, when a carbohydrate is attached to an extrinsic protein, a glycocalyx is formed, which is often used for cell communication and self-recognition to avoid an autoimmune response. Proteins in the surface membranes of cells can also act as receptor sites for hormones. They have a complementary shape to that of the hormone, and only appear in the membranes of the target cells. This is, for example, how mammary glands in marsupials are able to produce two sorts of milk. It also accounts for the specific action of reproductive hormones on certain organs. When a hormone receptor complex is formed, a second messenger, such as cyclic AMP, is formed. This can activate enzymes within the cell, and thus affect the cell's metabolism. Should the hormone be a steroid, it can enter the cell through the membrane, as it is lipid-soluble, forming a hormone receptor complex within the cell. The hormone ADH is released from the posterior pituitary gland. ADH targets the cells lining the second convoluted tubule and collecting duct in the nephron. When it binds to the complementary receptor sites on the membranes of these cells, aquaporins are stimulated to move from the cytoplasm to fuse with the cell membrane. This allows water in the filtrate to move rapidly into the interstitial fluid and therefore into the blood, avoiding dehydration. Also, in the gut, enzymes such as maltase are embedded in the plasma membrane of the cells lining the gut, which complete the absorption of carbohydrates. The membranes of these cells are folded to form large numbers of microvilli to increase surface area. There are also membranes present within cells. For example, the nucleus is surrounded by a double membrane with nuclear pores called the nuclear envelope. The pores allow entry and exit of substances such as mRNA. Mitochondria are also surrounded by a double membrane, the inner membrane of which is folded into cristae, and is the site of the electron transport chain, the last stage of respiration. Also, lysosomes are small membrane-bound vesicles formed from the Golgi apparatus, containing hydrolytic enzymes to break down unwanted organelles so the materials can be recycled. If the membrane did not surround these enzymes, they would destroy useful organelles, resulting in destruction of the cell. This essay has shown that there are many different functions of plasma membranes, with many different adaptations to these functions. How nitrogen-containing substances are made available and used by organisms. Nitrogen is found in every amino acid, protein, DNA and RNA. Most organisms, however, are unable to use atmospheric nitrogen directly. so the nitrogen cycle is very important. In this essay, I will describe how nitrogen is made available to different organisms and detail its many different uses. Atmospheric nitrogen, an unreactive element, is often fixed by nitrogen-fixing bacteria, such as Rhizobium, in the root nodules of leguminous plants to form ammonium ions that are released into the soil. Nitrifying bacteria then oxidizes the ammonium to nitrite and then to nitrate, which can either be denitrified to nitrogen or absorbed by plants through the root hair cells by active transport. Nitrogen may also be fixed by lightning, where the electrical energy allows nitrogen and oxygen to combine to form various oxides of nitrogen, which are then washed into the soil by rainwater, where they are taken up by plants as nitrates. Once plants have taken up nitrates, they can assimilate the nitrogen into its biomass and, on death, decomposer fungi can hydrolyze the proteins into amino acids and deaminate them to ammonium, which is released back into the soil for nitrification. Since the plant biomass can be consumed by primary consumers, the nitrogen can be cycled and recycled around an ecosystem, being made available for assimilation into biologically important molecules as it is transferred. Nitrogen is very useful to organisms, as it is one of the elements, along with carbon, hydrogen and oxygen, that is always present in proteins, in the form of an amine group. In a polypeptide, this amine group forms a peptide bond with a carboxylic acid group of another amino acid. These proteins can form enzymes which increase the speed of biochemical processes within cells, making nitrogen essential for many processes such as respiration and digestion. Nitrogen is also important for respiration in animals, as the oxygen used for this process is carried to the cells by red blood cells. It is the nitrogen-containing hemoglobin within these red blood cells that has a high affinity for oxygen. Nitrogen-containing membrane proteins also play a substantial role within the nervous system. These intrinsic proteins span the phospholipid bilayer of the axon plasma membrane and contain ion channels with gates which can be opened or closed to allow or prevent ions passing through. By actively transporting potassium ions into the axon and sodium ions out, these intrinsic proteins control the sodium-potassium pump, keeping the axon at resting potential of -70 mV until a nerve impulse generates an action potential. At resting potential, some of the potassium voltage-gated proteins are open, but those for sodium are closed, until opened by energy from a stimulus, allowing sodium to diffuse out, reversing the potential difference across the membrane. Once the potential difference releases +40 mV, these protein channels close, and the voltage-gated potassium channels open, allowing potassium ions to diffuse out, repolarizing the axon. Nitrogen-containing motor proteins also play an important role in muscle contraction. The proteins present are thin actin, consisting of two filaments twisted around each other, and thick myosin, which has bulbous heads. During contraction, the myosin head binds to the actin binding site and performs a power stroke, pulling the actin along. The ADP attached to the head is then released. This releases the head from the binding site. ATP then binds to the head and is hydrolyzed to ADP and phosphorus by ATPase, providing the energy for the myosin head to return to its original position. In this way, nitrogen is very important for the survival of organisms by aiding movement, so the animal may outrun its predator, or survive cold conditions by simmering, in which core muscles contract and relax, and by contracting hair erector muscles. Nitrogen also forms part of DNA in the form of nitrogenous bases, making nitrogen important in protein synthesis and the passing of genetic information from one generation to the next. Nitrogen can also form other compounds, such as NADP, which is used by plants during photosynthesis. During the light-dependent reaction, NADP is reduced to NADPH using hydrogen from photolysed water. The hydrogen transported by this molecule of NADPH is then used, along with energy from ATP, to reduce glycerate 3-phosphate or triose phosphate during the Calvin cycle of the light-independent reaction. Nitrogen is also often used in fertilizers by humans to increase crop growth. However, the additional nitrogen can leach into the ground and result in runoff entering rivers, causing eutrophication, as these nitrates promote growth of algae on the river, blocking the sunlight so the plants beneath cannot photosynthesize and therefore die. Bacteria break down these dead plants, using up oxygen as they do so. The lack of oxygen causes fish to die, which are also broken down, leaving the river putrid. This essay has highlighted how nitrogen is made available to organisms and the many different uses they have for it. How the structure of cells are related to their function. The cell is the simplest living unit of which all tissues, organs and organisms are built up. In multicellular organisms, these cells differentiate and become specialized to perform a given function. These specializations then impact on the structure of the cells. This essay will describe how the structure of some cells can relate to the cell's function. Species in the kingdom Plantae are anchored into the soil by their roots. The function of these roots include the absorption of water and mineral ions from the soil, which they deliver to the xylem for transport up and around the plant. The roots have an outer layer of cells called root hair cells that are adapted for this process. The cells have a long thin extension of the cell surface membrane, which helps with anchorage and, due to the dramatically increased surface area, has many channel proteins. Mineral ions such as nitrate and potassium ions are transported across the membrane by active transport using energy supplied by ATP. The ions pass through a specific hydrophilic channel protein against the concentration gradient. A higher surface area of the membrane means greater concentration of these channels, so a greater efficiency in absorption from the soil. These ions lower the water potential inside the root hair cell, allowing water to enter by osmosis. Here again, the presence of a greater number of hydrophilic channels offers the smaller water molecules an easy passage through the membrane. The root hair cells also have many mitochondria to help carry out respiration, providing the energy needed for this active transport. The water moves via the apoplastic and symplastic routes to the endodermis, where it enters the xylem tissue, the vacuolar route by which water passes up the stem of the plant. The xylem tissue is composed of xylem vessels that are tube-shaped hollow cells with pits in the side. They are hollow to allow an unhindered passage of water up through the plant. The absence of organelles further facilitates the passage of water. The pits in the side allow lateral passage of ions from the xylem out into the surrounding tissue to lower the water potential, allowing water to pass out and be used for support and, among other things, photosynthesis. The xylem cells themselves were originally cells that died due to a build-up of lignin in their walls. This makes them impermeable to water, so none is lost before being transported. Plants are a kingdom of species that are autotrophic, so they derive their energy through photosynthesis, by absorbing specific wavelengths of light and converting it into chemical potential energy, driving the combination of CO2 and water to form glucose and oxygen. The palisade cells situated mainly on the upper surface of the leaf and have many adaptations to help them absorb specific wavelengths of red and blue light. The palisade cells have a block-like shape that allows them to stick together with small gaps between them, much like a brick wall, to present a high surface area for light to strike, increasing efficiency. Each palisade cell has a large number of chloroplasts containing the green pigment chlorophyll, which absorbs the energy and uses it to excite electrons in the light-dependent reaction of photosynthesis. When their energy is released in the electron transport chain, ATP and NADPH are formed and used to reduce glycerate 3-phosphate to triphosphate and glucose. Animals are a kingdom of species that are heterotrophic. They derive their energy by consuming, digesting and absorbing plant and animal matter, which they use to provide nutrients, such as carbohydrates and lipids for growth and repair. The polysaccharide starch, formed from the glucose in plants, is consumed by herbivores and omnivores and digested in the gut by amylase to maltose. This disaccharide is then further hydrolyzed by maltase in the intestinal epithelium to glucose, which is absorbed by the epithelial cells located there. These epithelial cells have a brush border composed of microvilli, close-packed folded extensions of the membrane that massively increase the surface area and the concentration of channel proteins, through which glucose is absorbed by a sodium-glucose cotransporter. The many mitochondria inside these cells provide ATP for respiration, for the active transport of potassium ions from the blood into the cell, at the same time as transporting sodium out. This lowers the concentration of sodium in the cells, so sodium is drawn in, as is glucose. Glucose channels on the basement membrane then allow the glucose to enter the blood by facilitated diffusion. One role of glucose in animals is to supply a source of energy for aerobic respiration. This process requires the delivery of oxygen from the lungs to the respiring tissues. The oxygen is carried bound to hemoglobin inside red blood cells, small cells that have a biconcave shape. That means they can flex easily and offer a high surface area of contact with the capillary walls for efficient gas exchange. This reduces the diffusion pathway of the oxygen and carbon dioxide, increasing gas exchange rates. An absence of organelles also increases the room available for hemoglobin, each molecule of which can load 4 oxygen molecules. The respiration of this glucose provides energy in the form of ATP. This can be used to power the generation of a resting potential in a nerve cell. A nerve cell is adapted for the passage of electrical impulses from the CNS to effectors. Thread-like extensions of the membrane, called dendrites, allow the synapsing of information with other neurons, allowing the propagation of the action potential to more effectors. A long, thin, myelinated axon allows a direct connection to the CNS, along which waves of depolarization can pass. The rate of conductance is increased by the myelin, which insulates the membrane, allowing impulses to jump between the nodes of Rambia in saltatory conduction. The cell body of sensory neurons is located to the side to avoid interfering with the passage of impulses. Using DNA in science and technology. Deoxyribonucleic acid is made up of a number of nucleotides joined together by a double helix and carries the genetic code for all living organisms on the planet. Each of these nucleotides consists of a phosphate group, a molecule of deoxyribose sugar, and an organic base. Adjacent nucleotides are joined by a condensation reaction, forming the phosphate sugar backbone of a polynucleotide strand. 2 complementary strands are then joined by hydrogen bonds by specific base pairings, A to T and G to C, and wind together to form a double helix. On some occasions, the sequence of bases on a DNA strand may change by mutation. This may be spontaneous or can be caused by mutagens such as radiation. Mutations can be due to substitutions or deletions of bases. The changes in nucleotide sequence can lead to the section of DNA coding for a different polypeptide, resulting in genetic diversity. For example, in a bacterial cell, this mutation may code for antibiotic resistance, altering the frequency of alleles in the population. DNA base sequences may be compared by a process known as genetic fingerprinting, during which a sample of DNA is extracted from an organism and digested using restriction endonucleases, which cut certain phosphodiester bonds, leaving different length strands. These strands are then separated by gel electrophoresis, in which they are placed on agarose gel and voltage is applied. Differences in resistance mean smaller strands travel further. After this, DNA probes are binded to the core sequence and develop X-ray film to reveal a series of bars unique to the individual. These series of bars can then be compared from different organisms to see how closely they are related. This can be useful in comparing blood at a crime scene to that of a suspect in forensic science, or in a paternity test. DNA-based sequences can also be compared by DNA hybridization, in which DNA of 2 species are split into single strands by heating. Single strands from each species are then recombined as a hybrid. This hybrid is then heated. The higher the heat needed to separate the strands, the more closely the 2 species are related. DNA is also used in science and technology in the form of recombinant DNA, which is used to produce transformed organisms that benefit humans. In this process, a vector is required. The most commonly used vector is a plasmid, a small ring of DNA found in bacteria. Restriction endonucleases cut the plasmid, and DNA is incorporated that has also been cut by the same enzyme. The gene is fixed into place using DNA ligase. The plasmid is then replicated inside the bacterium, producing large amounts of the gene product. This can then be used to treat certain genetic diseases in humans, such as cystic fibrosis, where the required gene is inserted into the plasmid, replicated in the bacteria, and then introduced into the lungs through the nostrils, where it can insert the new gene into epithelial cells using a conjugation tube. This technology can also be used to increase the yield of plants or to introduce resistance to a disease. It can also be used to make vaccines. Gene therapy may be used in two possible ways: somatic cell therapy, in which the genes are inserted into the cells of the adult where they are needed, as described above, or germline therapy, where the genes of gametes or an early embryo are changed so that the offspring will contain the new gene. Another use of DNA technology is for genetic screening for disease. The order of nucleotides on the mutated gene is determined by DNA sequencing. A fragment of DNA with complementary bases to the mutated portion of the gene is then formed and is labelled radioactively, producing a DNA probe. PCR techniques are then used to produce multiple copies of this probe, which are then added to a single-stranded DNA fragment from the patient. If the mutated gene is present, some donor DNA fragments will bind to the complementary bases on the probe, so will expose X-ray film. How carbon dioxide affects organisms and ecosystems. An ecosystem comprises all the interactions between biotic and abiotic features in a given area at a given time. One key molecule that is of fundamental importance to these interactions is carbon dioxide, as it transfers carbon atoms between organisms and their environment. This essay will explore some of the important roles of carbon dioxide in a variety of organisms such as plants and animals. Today's atmosphere contains low levels of carbon dioxide, a gas that is a limiting factor for productivity of an ecosystem. Plants absorb their gas through their gas exchange surface, the stomata, on the underside of the leaves. It diffuses through the air spaces and into the stroma of the chloroplast, inside palisade cells, where it combines with ribose biphosphate to form two molecules of glycerate 3-phosphate, which is then reduced by NADPH and energy from ATP, both of which are formed in the light-dependent reaction, forming triose phosphate. This simple monosaccharide is then combined into larger molecules such as glucose, and ultimately the polysaccharides starch and cellulose. In this way, carbon dioxide acts as a source of carbon atoms, which were incorporated into the biomass of plants, and therefore the available biomass of an ecosystem. It follows that the higher the carbon dioxide levels in the atmosphere, the higher the productivity, but other limiting factors such as temperature, pollution, and availability of water and nutrients in the soil will also play a role. The biomass of the producers may then be consumed by herbivores, such as sheep, to provide energy and the building blocks for growth and repair. Starch formed by plants is digested by amylase to maltose, then by maltase into alpha-glucose. The glucose can then enter cells where it undergoes aerobic respiration, a process which provides energy in the form of ATP, but also releases CO2 back into the atmosphere. The glucose is oxidized to pyruvate, which is then systematically decarboxylated in the Links and Krebs cycle, in the matrix of the mitochondria. Decarboxylation releases CO2 and reduces NAD and FAD, from which ATP will be produced by oxidative phosphorylation in the cristae. Carbon dioxide is released as a waste gas and accumulates in the tissue fluids of the respiring cells. Here it plays a role in oxygen transport and delivery to respiring cells. Oxygen is bound to hemoglobin in the form of oxyhemoglobin. When the partial pressure of oxygen is low, for example when CO2 is formed by respiring cells, then the oxygen is unloaded from the hemoglobin in the red blood cells and is made available for respiration. The CO2 then replaces oxygen on hemoglobin and can be transported to the lungs, where it is returned to the atmosphere when expiration takes place. The CO2 also has some important physiological roles. An increased rate of respiration, for example during exercise, produces more waste CO2 at a time when oxygen demand is high. The CO2 dissolves in the blood plasma, releasing hydrogen ions which lower the pH. The reduced pH is detected by chemoreceptors in the walls of the carotid arteries and aorta, which send increased frequency of impulses to the cardioaccelerory center in the medulla oblongata of the brain. This in turn sends an increased frequency of sympathetic nervous impulses to the SAN in the wall of the right atrium, causing it to send an increased frequency of waves of excitation across the heart, increasing heart rate. This increases the cardiac output, resulting in faster delivery of blood to the gas exchange surface, the alveoli of the lungs. Thus, more oxygen can be transported around the body and more CO2 returned to the atmosphere. As all living things respire, the CO2 removed by plants for photosynthesis is constantly replaced. This constant recycling of CO2 is crucial to the existence of all life on Earth, as changing CO2 concentrations could have disastrous consequences for all life. Each organism is adapted by natural selection to exist optimally within its environment. Human activities such as deforestation and the burning of fossil fuels have resulted in an increase in atmospheric CO2. The COO bond can trap infrared radiation and prevent it from being radiated back into space. This extra trapped kinetic energy causes global warming, which is a large factor in the melting of the polar ice caps. This changes the environment for those adapted to living in Arctic conditions and causes sea levels to rise, which may result in the flooding of low-lying coastal land, increasing salination of soil, so these areas will begin to favour xerophytically adapted plants, as the soil has a reduced water potential. Xerophytically adapted plants will survive and pass on their genes, altering allele frequencies. Such huge changes in the environment will destabilise complex food webs and reduce diversity. While CO2 levels are only one factor in environmental change, the molecule's key importance to life make it essential that its levels are closely monitored and action is taken to minimise the various impacts of change. Causes of disease in humans. A disease is a disorder of a tissue, organ or system of an organism. There are many different types of disease, such as infectious disease, genetic disease, and autoimmune disease. This essay will detail the causes of some of these diseases. Infectious disease is caused by pathogens attacking an organism. These pathogens may be bacteria, viruses, protozoa, fungi, or parasites, and cause disease by gaining entry into the host, colonising tissues, and resisting the host's defences and causing damage to the cells. One disease caused by bacteria is cholera, where Vibrio cholerae in the faeces or vomit of human sufferers, or in contaminated water, enter the body and multiply in the intestine, where they secrete a toxin which increases the secretion of chloride ions into the intestine, causing the water potential to be lowered. For this reason, water leaves the cells by osmosis and enters the intestine, causing severe diarrhea and in turn dehydration. Another bacteria that causes disease in humans is Mycobacterium tuberculosis, which is spread via droplet infection and causes disease by producing lesions on the alveoli of the lungs, which results in epithelial cells growing as part of the body's response to this damage. These cells cannot perform gaseous exchange, reducing the efficiency of each breath and causing inflammation which leads to a painful cough. Bacteria that cause these diseases can transfer DNA from one bacterium to another by horizontal gene transmission, where one bacterial cell produces a conjugation tube from the cell membrane, which attaches to another bacterium, allowing a small ring of DNA, a plasmid, to be transferred. If the first has a gene mutation that codes for antibiotic resistance, this is spread to the other so both become resistant. As mentioned above, bacteria are not the only disease-causing pathogens. Fungi can cause infection known as mycosis, such as athlete's foot, which causes scaling, flaking and itching, as dermatophytes feed off of dead skin cells. Yeast, another fungus, can also cause candidiasis, also known as oral thrush, when naturally occurring fungi in the mouth build up due to poor oral hygiene, smoking, or a weakened immune system. Viruses also cause disease, such as HIV, by penetrating host cells, preventing the cell's normal metabolic processes, and using its nucleotides to replicate the virus DNA. The way in which we live our lives can also have a profound effect on our health. One example of this is atherosclerosis, a disease caused by a diet high in saturated fats. This circulatory disease develops when fatty streaks develop under the lining of the endothelium of a blood vessel, which develops into a fatty lump called an atheroma. The endothelial lining sometimes splits as a result of this, which can lead to a blood clot. A diet high in saturated fats may also cause coronary heart disease, in which the Coronary artery becomes partially blocked, reducing blood flow. If total blockage occurs, it can result in myocardial infarction. Other lifestyle choices that can result in disease include smoking, which also increases the risk of heart disease, as well as causing emphysema, where the elastin of the alveoli is permanently stretched, bronchitis, where bronchioles become inflamed, and lung cancer due to the carcinogens in tobacco, which can cause malignant growth to form. A diet high in sugar can cause type 2 diabetes, as the receptors on the liver lose their sensitivity to insulin. Type 1 diabetes mellitus, however, is due to an autoimmune disease in which the body's own immune system attacks the beta cells of the pancreas, preventing insulin production. Another cause of disease can be genetics, often as a result of a gene mutation where a base is replaced, deleted, or added, changing the base sequence. Diseases such as cancer are caused in this way. In normal cells, rate of division is controlled by proto-oncogenes, which stimulate cell division, and tumour suppressor genes, which slow cell division. If a mutation occurs, it may inactivate these tumour suppressor genes, resulting in uncontrolled cell division, which can lead to a malignant tumour. For example, a mutation of the p53 tumour suppressor gene prevents it from coding for the protein for apoptosis, the process by which cells kill themselves, and a mutation of the BRCA gene can lead to a high risk of breast cancer or ovarian cancer. These mutations can be spontaneous or due to an exposure to a mutagen, such as asbestos, which can be inhaled and cause mesothelioma, a cancer of the chest cavity. Another genetic disorder is cystic fibrosis, caused by a defective gene. The result is the epithelial membranes of the lungs being dry and producing viscous sticky mucus, causing congestion in the lungs and breathing difficulties. Damage to the nervous system can also cause disease. Examples of this include Parkinson's disease caused by damage to the extrapyramidal system, part of the motor nervous system concerned with muscle tone and maintenance of posture, and Alzheimer's disease caused by tangles in the microtubules of cells as a result of splicing failures. Some animals, such as snakes or spiders, can also cause nerve malfunctions by injecting venoms that act as neurotransmitter antagonists by binding to and blocking receptor sites on the postsynaptic membrane and preventing depolarization of the postsynaptic neuron, and in turn preventing muscle contraction. If the muscles that this affected were intercostal, it would result in breathing failures. Transfer of energy within and between organisms. All living organisms, such as plants and animals, require energy to power their cellular processes. This energy may be in the form of the molecule ATP or as heat. This essay will detail the processes by which energy is transferred inside and between organisms and how these transfers are utilized. In biological processes, the immediate energy source is often in the form of adenosine triphosphate. ATP is readily hydrolyzed by ATPase to ADP and an inorganic phosphate ion. This hydrolysis releases a packet of energy of approximately 31 kilojoules per mole, which can be used to provide energy for processes such as muscle contraction, active transport, or the generation of a nerve impulse. Plants are able to produce ATP during the light-dependent reaction of photosynthesis in the thylakoids of the chloroplast. Red and blue wavelengths of light are absorbed by chlorophyll on photostem 2. The energy absorbed is transferred to electrons and excites them to a higher energy level. This stimulates the photolysis of water, forming hydrogen ions, electrons, and oxygen. These electrons then replace those excited by light. The excited electrons are then passed down an electron transport chain in a series of redox reactions from one carrier to the next. The energy released by this passage is used to activate ATPase to combine ADP and PI to form ATP. The hydrogen ions released by photolysis are used to reduce NADP. The ATP and NADPH are then used for the reduction of glycerate 3-phosphate to triose phosphate in the light-independent reaction in the stroma of the chloroplast. This triose phosphate can be used to form glucose for respiration, stored as starch, or incorporated into other plant tissues. The glucose or starch can be used as an energy source directly by the plants, or indirectly by animals, to which the energy source is transferred when they consume, digest, absorb, and assimilate the sugars. Glucose, a 6-carbon monosaccharide, is a commonly used respiratory substrate. The glucose enters the cell through a specific protein channel by facilitated diffusion down a concentration gradient using only kinetic energy provided by heat. In the cytoplasm, each glucose is phosphorylated by 2 molecules of ATP, making it more reactive. The 2 triose phosphates formed are then oxidized and each release 2 ATP, becoming pyruvate and reducing NAD to NADH. Under anaerobic conditions, the pyruvate is reduced to lactate in the muscles, but in the presence of oxygen, the pyruvate enters the matrix of the mitochondria where it undergoes oxidative decarboxylation, forming acetyl coenzyme A. This joins with a 4-carbon intermediate and undergoes 2 further decarboxylation steps and oxidation, forming ATP, NADH and FADH. The energy from the original glucose is now stored in the electrons in the bonds of these reduced coenzymes. Upon diffusion to the cristae, these electrons are passed along an electron transport chain in a series of redox reactions. As the electrons pass down, protein channels in the membrane become permeable to protons, which then pass into the intermembrane space, lowering its pH. This in turn opens a channel that allows the protons to flood back across the membrane, generating an electrochemical gradient which activates ATPase, leading to the production of ATP. It's important to know that respiration processes also produce a lot of heat that is used to provide kinetic energy for processes such as osmosis and diffusion of molecules such as oxygen at exchange surfaces. As previously mentioned, the ATP is used to provide energy to a host of reactions in all organisms. In animals, hydrolysis of ATP provides energy for the detachment and formation of actomyosin crossbridges during muscle contraction. The Ratchet process allows actin filaments to be drawn into myosin, reducing the length of the sarcomere. In this way, an animal may run to avoid predators or catch prey, and the chemical energy from ATP has been converted into kinetic energy. Transfer of energy from ATP also plays a role in the transfer of nervous impulses along neurons to the muscles. ATP supplies the energy for pumping 3 sodium ions out and 2 potassium ions into the axon of a nerve against a concentration gradient. This resting potential of -70 millivolts is then maintained by reduction of the membrane's permeability to sodium ions. Once an action potential arises at a synapse or a neuromuscular junction, ATP again provides energy to move and fuse vesicles containing the neurotransmitter acetylcholine with the presynaptic membrane. Plants require the translocation of glucose as sucrose from source cells to sinks, such as roots, fruits and shoots, to provide energy to non-photosynthetic tissues. This is achieved through the active transport of sucrose, using energy from ATP, into the phloem, reducing the water potential. This causes water to enter by osmosis, generating hydrostatic pressure which forces the sugar solution up and down the phloem towards the sink regions. In order to provide support, plants require a flow of water to enter the roots from the soil. This is again achieved by active transport of mineral ions such as potassium and nitrate against a concentration gradient, using energy supplied by ATP. This lowers the water potential in the root hair cells, drawing water in, which moves towards the endodermis through the apoplastic gaps between cells and symplastic through cell cytoplasm pathways. In summary, this essay has shown how the energy in light is transferred to glucose in plants and then between organisms in many different forms. Why offspring produced by the same parents are different in appearance. Different offspring from the same parents inherit the same genetic information when the haploid gametes produced by the parents in meiosis fuse to form the zygote, which then divides to form the embryo. Despite this, they may have different appearances or phenotypes. This essay will explore some of the reasons behind these differences. The characteristics of an organism are encoded within its genes. Genes are sequences of bases on DNA which code for the sequence of amino acids in a polypeptide chain. Some proteins, as enzymes and gene expression factors, determine the structure and function of cells. They determine the development of an organism. While organisms of the same species carry the same genes at gene loci, the structure of these genes vary. These alternate forms of genes, or alleles, can give rise to the intraspecific variations seen within populations of the same species. Variation in alleles can be due to factors such as mutation, or through crossing over or independent segregation in meiosis. Mutations, such as substitution mutations, alter the base sequence of DNA, which in turn alters the complementary mRNA formed at transcription, and so the sequence of amino acids in the protein that would be coded for. They may occur randomly during the semi-conservative replication of DNA during later interphase of mitosis, or as a result of exposure to an environmental mutagen. X-rays and many estrogenic chemicals found in food packaging, for example. This can cause subtle alterations in the base sequence. If, for example, the mutated base sequence now codes for a non-functioning form of an enzyme that causes a darkening of a yellow pigment to brown in the coats of Labradors, then one offspring of 2 chocolate brown Labradors may appear pale yellow due to the presence of the mutation, whereas another puppy may be dark due to normal amounts of pigment being deposited in the fur. Mutations can be relatively rare, but meiosis is adapted to bring variation into a sexually reproducing population. Meiosis is the process of cell division that halves the chromosome number to produce haploid cells that are genetically different. This is an important process as it not only introduces genetic variation into a population, but also allows the restoration of the diploid number of chromosomes upon fertilization. During the first division of meiosis, homologous chromosomes line up with their partners and crossing over may occur. The homologous chromosomes join at points known as chiasmata, and sections of the chromosomes are exchanged, resulting in new combinations of maternal and paternal alleles. Each of the 4 new daughter cells formed will carry 1 copy of each chromosome. Before the chromosomes are separated into the cells, they align randomly at the equator of the cell. One of each of the 4 versions of the 23 homologous pairs is randomly and independently segregated into daughter cells, resulting in new combinations of maternal and paternal chromosomes. In this way, each gamete formed by both parents is genetically unique. And since the process of sexual reproduction is a random event, in that any sperm may fertilize the particular ovum released during that particular menstrual cycle, then no 2 offspring from the same parents can carry exactly the same alleles, since it is the combined effect of the alleles and the environment which ultimately determine the nature and appearance of offspring. No 2 will appear identical. The genotype of an organism is its genetic constitution, in other words, the alleles that make up each gene. For example, the gene for eye colour may have 2 alleles for brown being dominant and blue. 2 heterozygous parents, each carrying big B, small b, will each form gametes big B and small b, which could combine by random fusion during sexual reproduction, forming a variety of possible combinations, for example 2 dominant or heterozygous, which would give brown-eyed offspring, or homozygous recessive, which would give blue-eyed. As every gene has 2 alleles, there are a multitude of possible combinations of subtle differences in phenotype, each of which would give rise to a slightly different offspring. An organism inherits its alleles from its parents, but offspring from the same parents are rarely similar in appearance. Even identical twins with the same genotype can vary due to subtle differences in environmental factors that influence them in different ways. For example, each twin may be influenced by peer pressure to eat different foods or take part in different sports. Thus, one twin may appear leaner and have a more defined musculature than the other. The effects of childhood illness or conditions such as acne may also create differences in appearance later in life. Environmental influences are present in all species. One key factor in plants, for example, is the availability of key nutrients or energy. 2 dandelion plants from the same parents may differ in appearance if one has been grown in a shaded and dry field poor in magnesium. This plant would appear more yellow compared to one grown in magnesium-rich soil. While it is difficult to ascribe exact causes of subtle intraspecific variations, even those obvious in siblings of the same parents, some of the genetic and environmental reasons behind those differences are understood. The variation it creates is of key importance to the population, as it creates a large gene pool that has a greater ability to adapt and survive under more adverse and hostile conditions. The genetic and environmental causes of variation described here contribute to this diversity. The biological importance of water. Water is a chemical compound with the chemical formula H2O. A water molecule contains one oxygen and two hydrogen atoms joined by covalent bonds and is an important part of life. Without it, life on Earth would not exist. Water has many uses to organisms, some of which will be detailed in this essay. Water is a major component in cells, typically forming 70 to 90% of the cell's mass. In humans, water is around 80% of our mass. Water also provides an environment for organisms such as fish, as 75% of the Earth is covered in water. One biological use of water is as a solvent. Most substances found in the human body are soluble in water, apart from fats and other large polymers such as fibrous proteins. Substances that dissolve in water are known as hydrophilic substances and are usually polar or ionic. Water's property as a solvent is biologically important to life, as most biochemical reactions, such as respiration, occur in solution. It also allows water to be used as a means of transportation. Blood plasma is mostly water and is used to transport vital substances such as glucose around the body and sucrose in the phloem. Water also plays a role in many metabolic reactions. One example of this is hydrolysis, the addition of water in the breakdown of large biological molecules into their monomers, such as the breakdown of protein into amino acids. Water is also a product of respiration, produced when the hydrogen atoms from reduced coenzymes release their energy to form ATP, and then combine with oxygen to form water. This is especially useful to organisms living in dry habitats. Another metabolic reaction that requires water is photosynthesis. During the light-dependent reaction, light energy of certain wavelengths is absorbed by the chlorophyll. This energy excites an electron, which is then passed down an electron transport chain in a series of redox reactions. This light energy also supplies the energy for the photolysis of water, an important process as it replaces the excited electron, produces a molecule of oxygen which can be used for respiration, and releases a hydrogen ion which reduces NAD to produce NADH, which goes on to reduce glycerate 3-phosphate in the light-independent reaction, forming triose-phosphate, which goes on to form glucose. Water is not easily compressed and therefore plays an important role in support in plants and animals. Water enters plant root hair cells by osmosis after the active transport of mineral ions into the roots lowers the water potential. This water creates pressure against the cell wall, keeping it turgid. Water is also useful to plants due to its cohesive properties. The cohesiveness gives the water surface tension, holding droplets together. This means that as water evaporates from the leaves through the stomata, a transpiration pull is caused, pulling water up the xylem. This again aids in the transportation of soluble substances around the plant. The surface tension also allows aquatic insects to walk on the surface of water. Another biologically important use of water is in thermal regulation. Water has a high specific heat capacity, which means it absorbs a lot of heat energy for its temperature rise and loses a lot to cool. This helps to reduce temperature fluctuations in organisms, minimize increase in temperature in cells due to biochemical reactions, and reduce fluctuations in the temperature of aquatic habitats. A lot of heat is required to turn water into vapour, as it has a high latent heat of vaporization. This means that water helps some animals to maintain a constant body temperature, as a high amount of heat energy is removed from the body when sweat evaporates or during panting. It also has the same effect on plants as water evaporates from the leaves. This is important as it prevents the temperature becoming too high and enzymes denaturing. The importance of hydrogen bonds. A hydrogen bond is an interaction in which a hydrogen atom bridges two electronegative atoms. Hydrogen bonds are extremely important in biological systems. Their presence explains many of the properties of water. They stabilize and determine the structure of large macromolecules such as proteins, and they are involved in the mechanism of enzyme catalysts. This essay will detail some of their uses and why they are so important. The structure of DNA relies largely on hydrogen bonds between nitrogen-containing base pairs, found in every nucleotide. The double helix structure is made because of this, and is very compact and therefore efficient at storing information. Hydrogen bonds also help DNA form a large, stable molecule. If DNA wasn't large and stable, it would fit through the nuclear pores and leave the nucleus. Hydrogen bonds also allow easy replication and transcription, as they are easily broken by DNA helicase to separate the double helix into two template strands. Proteins also require hydrogen bonds to ensure a specific tertiary structure. Hydrogen bonds occur between alpha helixes and beta-pleated sheets, forming this tertiary structure of the protein. Without hydrogen bonds, these proteins would not form their required shape and would be unable to function. For example, the active site on an enzyme is formed using hydrogen bonds. If these bonds break, often due to high temperatures or a change in pH, the shape of the active site will change and the enzyme will become denatured and will no longer be able to form an enzyme-substrate complex, as the shapes will no longer be complementary. Hydrogen bonds are also responsible for the cohesion properties of water, making them very important in the survival of plants. The hydrogen bonds in water causes molecules to stick together, so as water evaporates from leaves, this causes a transpiration pool, pulling water up the xylem through the plant. Hydrogen bonds are also important to plants due to their presence in cellulose. Cellulose is a long chain of beta-glucose units joined by B14 linkages. These chains hydrogen bond together into fibres, giving cellulose its unique properties of mechanical strength and chemical stability. These cellulose fibres make up the rigid cell wall of plant cells, making hydrogen bonds important in maintaining the structure of plants. The effects on organisms of an increase in global temperatures. The rise in the environmental temperature can affect many organisms either in adverse or beneficial ways. The effects can lead to changes in the metabolic processes inside the organism, and over time can force adaptations and natural selection which may shape the interactions of the organisms within their niche. This essay will specify some of the effects and describe their ecological importance. Microorganisms such as bacteria and fungi do not have complex homeostatic mechanisms to control their body temperatures. An increase in environmental temperature will therefore have a profound effect on their metabolism. Enzyme activities will increase due to increased kinetic energy, increasing their rates of respiration. In turn, they are able to replicate more quickly as more ATP is made available for growth. Microbes occupy a variety of niches, including acting as decomposers. An increase in respiration rates and numbers of saprobiotic organisms will increase decomposition rates and carbon and nitrogen recycling. Their carbon recycling releases more CO2 as a waste product of aerobic respiration, which could further add to global warming, as CO2 is a greenhouse gas. As many microbes are pathogenic, the increased growth rates will also increase the mutation rate, possibly leading to the production of more pathogenic forms. The antigenic variability that results from the change in gene sequence will allow some of the forms to evade the host immune system of the animal they infect, increasing the incidence of disease. Many single-celled photosynthetic algae will also have their growth rates increased in the same way. This increased nitrate recycling caused by the growth of nitrifying bacteria will allow greater leaching of nitrate into the water courses. This nitrate is a limiting factor in the growth of algae across the surface of the water. The algae will bloom, covering the surface and outcompeting aquatic plants for light, causing their death. This will in turn lead to rapid growth of the numbers of decomposers which aerobically respire, reducing the oxygen levels, causing fish to die, and allowing the proliferation of anaerobic organisms. This eutrophication process will dramatically reduce the diversity index and destabilize food webs associated with this ecosystem. Plants are the main producers for food webs in an ecosystem. They are autotrophic and photosynthesize to produce energy in the form of carbohydrates. The passage of water through the plant is called transpiration, and this process will be accelerated at higher temperatures. Plants obtain ions and water from the soil by active transport of mineral ions. This lowers water potential inside the root hair cells, causing absorption of water by osmosis. The water then passes through the cortex via the apoplastic and symplastic pathways and enters the xylem, where it ultimately causes a root pressure, which pushes water up the xylem. Evaporation of water from the mesophyll cells and its loss through the stomata creates a water potential gradient that draws water from the xylem to replace that was lost. Higher temperatures would mean a faster rate of transpiration, and so over time would force xerophytic adaptations through natural selection, such as reduced surface area to volume ratio, as seen in cacti, rolled leaves, and sunken stomata. Such changes are likely to reduce biodiversity of plants, as fewer species are adapted for hotter and drier climates. In turn, this would reduce productivity and the range of plants in a given area, meaning fewer habitats and fewer food sources for animals. The drop in productivity and diversity index would therefore lead to a destabilisation of food webs, which would lead to dramatic reductions in population numbers, creating genetic bottlenecks. These bottlenecks result when a large reduction in population reduces the range of alleles in the gene pool. The surviving population is less able to adapt to the new conditions, and so this increases the chance of extinction, which would further destabilise the remaining food webs. Endothermic mammals too would experience physiological effects of temperature rises. Thermoreceptors on the skin would be activated more and send a greater frequency of impulses along sensory nerves to the heat loss centre of the hypothalamus of the brain, which would in turn send more signals to the effectors, increasing the rate of sweating and vasodilation, and decreasing metabolic rate. Such longer-term physiological changes would therefore impose selection pressures that would select for less body hair and a leaner body with a higher surface area to volume ratio, to dissipate heat more effectively. One other physiological effect may be to alter the position of the oxygen dissociation curve of haemoglobin. A shift to the left would mean that less oxygen becomes unloaded due to an increased affinity as the rate of respiration is reduced. Ectothermic reptiles, such as lizards, would be active for more of the daytime, and their behavioural adaptations, such as basking, would help maintain their metabolism at optimum conditions. These organisms derive their energy mainly from their surroundings, and a warmer climate would increase their chances of competing with endotherms. Once again, the shift in ecological balance could have profound consequences for the food webs and affect the species diversity and the chance of survival of many struggling populations of organisms. Increased global temperature would also lead to the reduction of habitats such as the polar ice caps, which would melt, reducing the environment available for animals adapted to cold conditions, such as polar bears. Anatomical adaptations, such as thick white fur and large amounts of subcutaneous fat, acting as thermal insulation, will no longer be of benefit, so may be outcompeted by those that are smaller and darker. Enzymes and their roles in functions of cells, tissues and organs. Enzymes are biological catalysts that control almost all chemical reactions inside and outside of cells. In this way, they control the functions of not only individual cells, but of collections of cells or collections of tissues. This essay will demonstrate the diverse range of ways that enzymes contribute to the functioning of these structures. Enzymes are globular proteins that have a specific tertiary structure that has a complementary shape to that of a specific substrate molecule. The lock and key model is used to describe enzyme action. For example, the enzyme lactase has an active site that is complementary only to lactose. Sucrose, a similar disaccharide, has a different shape to lactose, so cannot bind to lactase's active site. On binding to the active site, an enzyme-substrate complex is formed and reaction takes place. This reaction can either be catabolic, in which the substrate is broken down into smaller units, or anabolic, in which smaller units construct a larger molecule. The products have a different shape, so detach from the active site. In the induced fit model, the active site is not complementary to the substrate, but on binding, the shape changes and the active site forms, moulding itself to the substrate. Humans gain the molecular building blocks they need for energy and growth from digestion of food by the digestive system. This is a system of organs that is adapted for the hydrolysis of food molecules and the absorption of their products. In the mouth, the enzyme amylase in the saliva hydrolyzes starch to the disaccharide maltose, which is further digested in the intestinal epithelium to alpha-glucose. In the stomach, endopeptidases such as pepsin break down proteins into smaller peptides, and exopeptidases such as trypsin further hydrolyze these into amino acids in the small intestine. Glucose is then absorbed by sodium glucose transport, a type of active transport that involves the enzyme ATPase, which hydrolyzes ATP to ADP and PI, releasing energy to pump sodium out and potassium into epithelial cells, creating a diffusion gradient for sodium and glucose uptake. Enzymes also play a key role in the digestion of large insoluble food molecules into smaller, more soluble products that can be transported and assimilated in fungi and bacteria. These decomposers release hydrolytic enzymes such as lipase, carbohydrase, and protease. The soluble products of the extracellular digestion can then be absorbed and assimilated into useful compounds. All organisms carry the genetic code for these functions as a DNA molecule. Before a cell divides by mitosis, the DNA must undergo semi-conservative replication to produce two identical copies for the daughter cells. Enzymes play a key role here. Helicase binds to the DNA, breaking the hydrogen bonds that hold the polynucleotide chains together. This reveals two template strands with exposed bases which bind to DNA nucleotides. DNA polymerase forms a phosphate sugar backbone by joining adjacent nucleotides with a condensation reaction. Some cells, such as beta cells of the pancreas, synthesize protein hormones such as insulin. In order for the genetic code on DNA to be expressed and the insulin to be formed, the DNA must be transcribed as pre-mRNA, spliced to form mRNA, and translated as a protein. Enzymes are involved in each step. Helicase binds to the gene locus, causing it to unwind, exposing the template strand. RNA polymerase joins adjacent nucleotides in a condensation reaction to form pre-mRNA. Enzymes in the nucleus remove non-coding introns and splice together the exons, leading to the formation of mRNA, which binds to a ribosome on the rough endoplasmic reticulum. tRNA complexes line up with their anticodons on the codons on mRNA and bring two amino acids in contact with an enzyme on the ribosome that condenses them together, forming a peptide bond. This process is repeated to build up the primary structure of the insulin molecule. The action of the hormone insulin also involves phosphorylase enzymes, which cause the condensation of glucose into glycogen in the liver by glycogenesis. All living cells release the energy in substrate molecules using aerobic or anaerobic respiration. The respiratory process is a sequence of interconnected enzyme-controlled steps, called a metabolic pathway. Other pathways include photosynthesis and the synthesis of steroid hormones such as estrogen from cholesterol. During glycolysis, the link reaction, and the Krebs cycle, some of the steps include oxidation by dehydrogenase enzymes. This oxidation involves a transfer of hydrogen ions and electrons from the substrate and passing them to a coenzyme, which becomes reduced. For example, in the cytoplasm, when triphosphate molecules are oxidized to pyruvate, the coenzyme NAD is reduced to NADH. This coenzyme forms part of the active site, and dehydrogenase allows it to function as a catalyst and be reformed. The ATP formed as a part of respiration is used in a wide variety of contexts in biology. For example, in order for an animal to move and hunt for food within its environment, it has to contract its muscle tissue. This tissue is composed of cells containing actin and myosin filaments, which move relative to each other to contract a sarcomere. For this to happen, actin-myosin cross bridges form between the actin and myosin. Once activated by calcium ions, ATPase hydrolyzes ATP to ADP and PI, releasing energy for the detachment and formation of more cross bridges, giving rise to the sliding filament theory. This enzyme also helps release energy from ATP in a wide variety of contexts, such as the active transport of sodium-potassium in the sodium-potassium pump, in the generation of a resting potential, or in the active transport of nitrate ions into the roots of plants to lower water potential to drive in water, producing a root pressure. Receptors and their role in coordination. Receptors are structures that detect specific stimuli and transduce their energy into nerve impulses as a form the body can interpret, allowing the coordination of an appropriate response. Receptors can detect external stimuli such as light, temperature, chemicals and pressure, or internal stimuli such as electrical, chemical, thermal, pressure or osmotic. This essay will discuss the variety of receptors and outline how each plays its role in coordination. Simple organisms such as woodlice are prone to dehydration. They are able to detect threatening decreases in the non-directional stimulus such as humidity via receptors. This triggers a kinesis, which causes an increase in random movement until they enter a more favourable environment. usually a darker, moister area. Single-cell algae are photosynthetic and have pigments that act as receptors, allowing the algae to respond to directional stimulus such as light. On detection, it causes a positive phototaxis, which moves it towards the light for photosynthesis. Other autotrophic organisms, such as plants, are phototrophic and grow towards light. Here again, the pigment chlorophyll in chloroplast of palisade cells is acting as a light receptor. For larger mobile organisms to move and coordinate effectively in their environment, it is an advantage if it can detect and make sense of light energy. In animals, the receptor surface is the retina of the eye, which comprises the receptor cells, rods and cones. The cone cells are located primarily in the centre of the retina, known as the fovea. Each contains one of three kinds of the pigment iodopsin, which will bleach on absorption of a specific wavelength of light. This bleaching causes a chemical change which leads to changes in the permeability of the attached bipolar cell's membrane to sodium ions. This causes the depolarization of the bipolar cells, which initiates a nerve impulse in the optic nerve. Each cone cell has a single attachment to the optic nerve and so sends large amounts of information to the sensory areas of the cerebral hemispheres, which can process the information and create vision of high acuity. The rod cells are more abundant at the periphery, and many are connected to one bipolar cell. This causes summation of signals and increases the chances of a generator potential being formed at lower levels of light at the expense of visual acuity. In this way, information about the environment is detected and passed on to the brain to be processed to create a visual image of the world around us in a variety of lighting conditions. In moving through its environment, the animal will be in contact with the ground and be touching and sensing constantly. For example, on touching a tree, Pacinian corpuscle receptors under the skin of a bear will detect changes in pressure. Stretch-mediated sodium channels will open, allowing sodium to enter and depolarize the axon of the attached sensory nerve. The information can then be processed by the brain. The animal may also encounter changes in temperature. In order to maintain the homeostasis of its internal environment, it would have to respond either by increasing or decreasing metabolic rate and initiating mechanisms to lose or gain heat. Temperature receptors on the surface of the skin can detect warm or cold conditions. For example, on activation, cold receptors establish a generator potential and send impulses to the heat gain center of the hypothalamus. As a result, this increases the frequency of impulses to the effectors such as muscles in the arterioles close to the skin, causing vasoconstriction, and the contraction of erector muscles on the skin, causing hair to stand up and providing insulation. Another response to low temperature is the onset of shivering, uncontrollable muscle contractions in the core that increase the rate of respiration and generate more heat internally. However, this will release CO2 as a waste product and increase the oxygen demand of respiring tissue. To compensate, heart rate must increase to deliver more blood to the lungs for gas exchange. The CO2 dissolves in the blood plasma and releases hydrogen ions, which lower pH. This is detected by chemoreceptors in the aorta and the carotid artery, which send impulses to the cardioexcitatory center in the medulla oblongata. In turn, this sends an increased frequency of impulses by the sympathetic pathway to the sinoatrial node in the heart, to increase the rate at which it sends waves of excitation across the atria. The heart can also respond to changes in blood pressure. If blood pressure is higher than normal, baroreceptors in the aorta send impulses to the cardioinhibitor center, which increase the frequency of parasympathetic impulses to the sinoatrial node, reducing the frequency of contraction. Using these autonomic pathways, the body is able to maintain its constant internal environment. Internal coordination may also be hormonal in nature, and here chemoreceptors play a role. Chemoreceptors are often protein molecules with a specific tertiary structure complementary to the shape of a certain chemical stimulus. This ensures only one specific stimulus is responded to. When starch is taken in as food and digested by the enzymes amylase and maltase into glucose, the glucose is absorbed into the blood plasma by a sodium-glucose cotransport. The increased glucose concentration causes the blood-water potential to be reduced, which may prove harmful. The increase in glucose is therefore detected by a glucose receptor on the surface of the beta cells of the islets of Langerhans in the pancreas. The receptor triggers the release of insulin, which travels to the liver where it binds to another receptor on the cell surface membrane of hepatocytes, forming a hormone receptor complex. This alters the shape of the receptor, triggering the activation of phosphorylase enzymes, which condense glucose to glycogen in glycogenesis, lowering blood glucose concentration. Chemoreceptors are also important in preventing disease. Phagocytes, which circulate in the blood, have receptors on their surface that are complementary to the surface of pathogens, such as bacteria or viruses. On binding, the process of phagocytosis is initiated, which leads to the engulfing of the pathogen and its subsequent destruction in the phagocytic vesicle by lysosomes. Presentation of this antigen on the surface of the cell allows a binding and recognition by B and T lymphocytes, which can further coordinate a more specific response to the infection. Lipids in health and disease. Lipids are molecules containing carbon, hydrogen and oxygen, and sometimes phosphorus, which play important roles in health and disease. This essay will examine some of these functions and look at how they contribute to health and disease. Triglycerides are lipids composed of a glycerol head group linked by an ester linkage to three fatty acid tails formed by condensation reactions. The fatty acid tails can be saturated in that they contain only single carbon-to-carbon bonds, or unsaturated with at least one double. The tails can be up to 17 carbon atoms long, giving rise to the molecule's very hydrophobic properties. This hydrophobic nature makes them insoluble and so good candidates for storage functions. Lipids have a lower density than water, which also make them good thermal insulators. Many animals have a thin layer of saturated fat under the skin, which helps to minimize heat loss through radiation. This means that they contribute to an animal's ability to maintain its body temperature by homeostasis. This is more pronounced in animals that are adapted to Arctic conditions. Often these animals are much larger and have a thicker layer of fat beneath the skin. The increased size this gives lowers the surface area to volume ratio, resulting in a lower effective surface from which heat would be lost. Coupled with more insulation, this means the animal would need a lower respiration rate in order to survive, thus helping to preserve fuel sources. The dense number of carbon-to-carbon and carbon-to-hydrogen bonds in lipids makes these molecules energy-rich, and so they act as useful fuels. A fuel is any substance that releases energy in a useful form when reacted with oxygen. Hydrolysis of triglycerides by lipase enzymes in adipose tissues releases fatty acids, which can be converted to glucose in the process of gluconeogenesis. In healthy individuals, carbohydrates are the usual source, unless exercise reduces the blood glucose concentration, resulting in hypoglycemia. In such situations, gluconeogenesis represents a mechanism by which new glucose can be synthesized to provide energy in the liver from glycerol. This is also useful in situations such as diabetes, where a sufferer may become hypoglycemic if they take too much insulin or do not replace glucose when exercising. Fatty acids can be converted to glucose, enabling a diabetic to make more glucose available despite having limited glycogen stores. The energy-rich lipid molecule therefore represents a key component of any diet. They not only provide energy, approximately 9 kilocalories per gram, but can dissolve certain fat-soluble vitamins which allow them to be absorbed and transported through the body. In this sense, they are key to the health of an individual, but too much intake in the diet can have a profound impact on health. Obesity increases the risks of coronary heart disease and myocardial infarction through the formation of an atheroma, a fatty deposit on the inside wall of an artery. In the coronary artery, these can lead to reduced blood supply to the heart, resulting in the death of muscle cells through lack of oxygen, causing myocardial infarction. Restricted blood flow can also contribute to high blood pressure, the risk of stroke, and other circulatory disorders. Obesity is also an important factor in the development of type 2 diabetes. However, unsaturated fats, such as those found in nuts and seeds, can be a healthier alternative to saturated animal fats. Plants can grow from nuts and seeds, and these contain the genetic information for the plant's development. They are also a rich source of unsaturated fats which act as the energy source for the germinating seed. Another type of lipid is a phospholipid. These molecules are similar to triglycerides but have one of the fatty acids substituted by a phosphate ion. This ion gives hydrophilic properties to the head, which combined with the hydrophobic tails gives these molecules a unique property of being able to align to form a phospholipid bilayer, the basis of the cell membrane. In the bilayer, the hydrophobic tails can align, producing a structure with hydrophilic properties on either side, which can interact with the aqueous environment, inside and outside of the cell. The molecules can move relative to each other, forming the fluid aspect to the fluid mosaic model. The mosaic aspect is related to the embedding of other structures such as glycolipids. These are lipids with carbohydrate structures attached that can act as recognition sites for antigens on the surface of bacteria such as Vibrio cholerae. In this way they can contribute to the ineffectivity and pathogenicity of some viruses and bacteria in that they represent a means by which these cells or their toxins can enter the host cell to infect them and cause disease. In the case of Vibrio cholerae, binding of a toxin to glycolipids attached to chloride ion channels can cause cholera, resulting in life-threatening dehydration. Also embedded within the bilayer is another type of lipid, cholesterol. This ring-like lipid not only adds strength to the membrane, but also acts as a precursor molecule to synthesize other related substances such as estrogen and progesterone, sex hormones involved in the control of the female's menstrual cycle. Estrogen is a steroid that stimulates the uterus lining to thicken, while progesterone helps maintain the thickened lining. Estrogen is also involved in a negative feedback loop with the protein hormone FSH, which causes the follicle to mature the developing ovum. The ovum releases estrogen, which inhibits the release of FSH from the pituitary gland. This feedback is exploited in contraceptive pills, where elevated estrogen levels prevent FSH maturing the ovum. Another important role for a lipid is the acceleration of nerve impulses in sensory and motor neurons of animals. A lipid, myelin, wraps around the membrane of nerve cells, forming an insulating sheath around the axon. Action potentials cannot form in these regions, so impulses jump between nodes of Ranvier by saltatory conduction, increasing the speed of conduction. In some diseases, such as motor neuron disease, the sheath peels away, resulting in slower conductance of impulses along nerves. Deoxyribonucleic acid carries the genetic code for all living organisms on the planet. It is variation in the information it carries in the form of genes and alleles that produces the wide diversity of life and the variations within and between species. This essay will describe the structure of DNA and illustrate the ways in which information encoded within it is transmitted within a cell and between cells and organisms. DNA is a polymer, a double helix of two polynucleotide strands bonded together by hydrogen bonds. Each nucleotide comprises a phosphate group attached to a 5-carbon deoxyribose sugar and an organic base containing nitrogen. These bases can be adenine, thymine, cytosine, or guanine. Adjacent nucleotides are joined by a condensation reaction to form the phosphate sugar backbone of a polynucleotide strand. Two complementary strands then join by specific base pairings, A to T and C to G, which then wind together to form the double helix, which provides strength and stability to the molecule. The information in DNA is encoded in the sequence of bases along the template strand of the DNA. A gene is a sequence of bases on DNA that codes for the sequence of amino acids in a polypeptide chain. Since proteins determine the function and structures of cells, it is the DNA code that controls all cellular activities. Organisms of the same species carry the same genes at fixed points called loci, but individuals carry different versions called alleles. Variations in these alleles result in intraspecific variation within a species. In order for the genetic material to be transferred into daughter cells, as the organism grows or repairs, DNA must be replicated by semi-conservative replication. The enzyme, helicase, binds to the DNA, breaking the hydrogen bonds, allowing the exposed bases on the two template strands to be revealed. DNA nucleotides then bind to exposed bases by specific base pairings with hydrogen bonds. DNA polymerase then join the adjacent nucleotides with a condensation reaction, forming the phosphate sugar backbone. Each of the two new DNA molecules formed contains one of the original strands of DNA, making the replication semi-conservative, which helps to minimize the incidence of mutations in the copying of the code. In prokaryotic cells, the DNA is free in the cell's cytoplasm, but in eukaryotes it is bound within a nucleus and joined to structural proteins called histones. The structure formed is called a chromosome, and it is these that must be separated for the daughter cells to carry the same genetic information as the parent cell. The process of mitosis separates the two copies of each chromosome. During prophase, the chromosomes coil up and become visible. The nuclear envelope disappears and the chromosomes attach to the spindle fibers at the equator of the cell using their centromere in metaphase. In anaphase, the centromere divides and the spindle contracts, drawing the chromatids to opposite poles of the cell. After telophase and cytokinesis, two new daughter cells are formed, each containing an identical copy of the DNA code, hence the encoded information has been transmitted vertically. Bacteria also possess the ability to transmit some of their genes horizontally. Conjugation tubes can form between two bacterial cells, and a plasmid can pass from one to another. If the bacterial cell has a plasmid that carries the code for penicillinase, this can be passed to another, so both cells will be resistant to penicillin. The information on DNA is encoded as triplets of bases called codons. Each triplet can code for one amino acid in a polypeptide chain. For example, GCA codes for alanine and TAC codes for glycine, so GCA-GCA, TAC-GCA would code for the polypeptide sequence ALA-ALA-GLY-ALA. As there are over 20 different amino acids and the triplet codes for up to 64, the code is degenerate, in that some codons code for the same amino acid. This minimizes the effect of mutations by allowing some to be silent, in that a different base is present but the same amino acid is coded for. Each of the codons is translated in sequence as the code is non-overlapping. But first the genetic information must be transcribed and then transferred out of the nucleus. It is transferred as an RNA molecule, a single-stranded polynucleotide containing the base uracil instead of thymine and a 5-carbon ribose sugar. Transcription produces a copy of a gene in the form of mRNA. Helicase binds to the gene locus, causing DNA to unwind and reveal a template strand. RNA nucleotides bind by specific base pairings, and RNA polymerase joins them by condensation reaction to form a strand of pre-mRNA. Introns are then removed and exons are spliced together with enzymes to form mRNA, which is small enough to diffuse through the nuclear pore and bind to a ribosome on the rough endoplasmic reticulum. The process of protein synthesis, translation, can now begin. In the cytoplasm, a tRNA molecule binds to a specific amino acid using amino-acyl RNA synthetase enzymes, forming amino-acyl tRNA, two of which deliver their amino acids to the ribosome, as the anticodon on tRNA binds to a complementary codon on mRNA. An enzyme then forms a peptide bond between the amino acids by condensation using energy from ATP. This process is repeated, building up the polypeptide chain. Alterations of the base sequence of the gene, mutations, therefore alter the polypeptide sequence. Sexually reproducing organisms transmit their genes in the form of haploid gametes formed by meiosis. This reductive cell division halves the chromosome number, so the diploid number of chromosomes can be regenerated on fertilization. Meiosis introduces variation through crossing over and independent segregation of chromosomes, and random fusion ensures further variation in offspring produced. In this way, the genetic information is transmitted from generation to generation, introducing a diverse range of alleles that adds not only variety, but helps to ensure a population can survive and adapt to environmental changes.